Thank you for the kind introduction and for the opportunity to present on the CARB Research Program. My name is Thushi Amini, and I am the Associate Director for Research in the Office of Infectious Diseases. I do not have any conflicts of interest to disclose. I would like to go over a few housekeeping notes. Please submit questions via the webinar's Q&A pod. Questions should be relevant to the topic addressed today. There will be a few minutes to answer questions at the end of each section, and the slides that I'm presenting today are intended as a basic summary. So please refer to the official funding announcement for more detailed information. My presentation today will begin with an overview of the CARB program, the ongoing studies that we have awarded, opportunities for collaboration, as well as where to find the information that I'm going to present. As Laura mentioned, the Office of Infectious Diseases is one of the clinical offices within the Office of New Drugs, which I've highlighted with the green arrow, within the Center for Drug Evaluation and Research in FDA. The Office of Infectious Diseases consists of three divisions: the Division of Anti-Infectives, the Division of Antivirals, and the Division of Pharmacology Toxicology for infectious diseases. I have highlighted the Division of Anti-Infectives because this division is responsible for protecting the public health by assuring that safe and effective drugs are available to the U.S. population for antimicrobial products. The public health impact is, as bacteria and fungi continue to develop resistance, standard treatment can become ineffective and bacterial and fungal infections threaten global health. Therefore, there is an urgent need to develop new antimicrobial drugs that are active against pathogens associated with antibacterial and antifungal drug resistance and poor clinical outcomes to improve patient health and well-being worldwide. This past month, on October 20th, the National Action Plan for Combating Antibiotic-Resistant Bacteria, or CARB, was renewed for another 5 years. The National Action Plan for CARB coordinates the strategic actions that the U.S. government will take in the next 5 years to improve the health and well-being of Americans by changing the course of antibiotic resistance. This plan is based on the U.S. government's 2014 National Strategy for CARB and builds on the first National Action Plan, which was released in 2015. This new plan describes activities that the U.S. government will undertake from the years 2020 through 2025 to reduce the impact of antibiotic and antimicrobial resistance in the United States. And what is unique now is that the new plan includes antifungal drugs under the term antibiotic to describe both antibacterial and antifungal drugs, which kill bacteria and fungi, respectively. I have provided the link to the updated CARB National Action Plan if you'd like to read more about it. The Office of Infectious Diseases receives annual funding from Congress to support CARB-related regulatory science research, and our studies focus on identifying regulatory science research needs to facilitate the development of new drugs that are active against multi-drug resistant bacteria and fungi. Therefore, the purpose of the CARB initiative in the Office of Infectious Diseases is to facilitate the development of new antibacterial and antifungal drugs to treat patients, and to advance the science of clinical trial design. The goals of the CARB research program are to help facilitate the development programs for antibacterial and antifungal drugs where limited resources or a lack of incentives is preventing the development of new antimicrobial drugs, and to create new drug development tools or standards that are publicly available for use by industry or other stakeholders to meet patient needs. The CARB research program aligns with HHS's strategic plan, FDA's strategic plan for regulatory science, and CDER's research goals. The specific CARB research areas of interest are to promote innovations in clinical trial design for new drugs, to advance the science of in vitro, animal model, pharmacokinetic studies, and real-world evidence studies to facilitate drug development, as well as advance the science of antibacterial drug susceptibility testing, also called breakpoints. And these topic areas are listed in FDA's Broad Agency Announcements section 2.4, which are specific for the CARB research program. I would also like to give you a high-level overview of the diversity of our ongoing studies that we're funding and the types of funding mechanisms we use to award our research studies. In fiscal year 2017 through 2018, we awarded several research contracts for the development of animal models of bacterial pneumonia caused by Pseudomonas aeruginosa and Acinetobacter baumannii. These pathogens were listed in the first tier group in the 2017 World Health Organization's list of antibiotic-resistant priority pathogens, where new antibacterial drugs are urgently needed. CDC also listed them in their antibiotic resistance threats report. So we awarded several contracts through FDA's broad agency announcement to develop both small and large animal models. 3 mice models, a catheter-associated bloodstream infection model challenged with Acinetobacter baumannii, and 2 pneumonia models challenged with Acinetobacter baumannii and Pseudomonas aeruginosa were awarded. In addition, we awarded a rabbit model contract because this larger animal reflects the human pattern of infection. The rabbit lung anatomy is similar to humans, and rabbit models reproduce many hallmark clinical features of hospital-acquired pneumonia and ventilator-acquired pneumonia. The purpose of these studies are to refine an existing model or develop a new animal model and then validate these animal models for testing investigational antimicrobial drugs in order to predict whether a drug will be efficacious in humans. In FY19, we awarded a task order contract under an existing IDIQ contract to determine the benefits and limitations of natural language processing. This proof of concept study analyzes data in unstructured notes in the electronic healthcare records and then assesses whether we can train a machine to recognize whether certain events took place. Also, in FY19, we awarded a grant to develop a patient-reported outcome, or PRO, instrument on symptoms from patients with non-cystic fibrosis bronchiectasis with or without non-tuberculosis mycobacterial lung infection. This project will conduct qualitative research to develop a content-valid draft PRO. This will be followed by a quantitative non-interventional study to validate the draft PRO instrument to a stage of readiness for future interventional trials and final qualification for drug development. We also have funded several multi-year interagency agreements with the National Institutes of Health, the Center for Disease Control and Prevention, and with the Department of Health and Human Services. The economic study with HHS is to identify how recently approved antimicrobial drugs are performing relative to their clinical value, the costs, and market returns, and contributing factors to this performance. This project explores the current and potential future burden of antimicrobial resistance, including the potential for better diagnostics to mitigate the current and future burden. Finally, this project will assess whether there are market failures for antimicrobial drugs that lead to suboptimal allocation of societal resources to developing antimicrobial drugs and options to mitigate these market failures. We have another ongoing study with CDC to understand the effects of antibiotics on the gut microbiome. The human microbiome, which is the normal bacterial colonization of the human body, including the gastrointestinal tract, the respiratory tract, and skin, protects us from disease. However, a common side effect of antibiotic use is microbiome disruption, where antibiotics can change the bacterial populations to make it a more favorable environment for multi-drug resistant organisms to grow and spread. The CDC study will identify and validate biomarkers of microbiome disruption in a microbiome model to measure antimicrobial disruption of the GI and in the respiratory tract. The third interagency study we have is with NIH, and it evaluates breakpoints for various bacterial drug combinations. Breakpoints identify whether bacteria are considered susceptible or resistant to a particular antibacterial drug. Bacteria can change over time, which may affect their susceptibility to antimicrobial drugs, which then may require breakpoints to be updated. NIH is helping us evaluate gram-negative bacteria and drug combinations in the CERNER database to determine if a relationship exists between minimum inhibitory concentration, or MIC breakpoints, and clinical outcomes, and whether there is an increase in mortality across a range of MIC values. In FY20, we will continue to develop 2 rabbit animal models of ventilator-associated bacterial pneumonia caused by carbapenem-resistant strains of Pseudomonas aeruginosa and of Acinetobacter baumannii. Another high priority for funding under CARB is to advance the science of antibacterial drug susceptibility testing to ensure that up-to-date breakpoints are available for patient care and antimicrobial stewardship. Under Section 3044 of the Cures Act, a system was created to expedite the recognition of breakpoints so that up-to-date information is available to the healthcare community in a streamlined manner so that physicians are able to select appropriate antibacterial drugs. And we awarded 4 contracts to determine whether breakpoints need to be adjusted, and these contracts were just recently awarded last month. So I also wanted to point out the timeline for the CARB research studies. They can be anywhere from 1-year studies to multi-year studies, and these depend on the scope of work being performed. So based on public input, for example, from FDA workshops, as well as input from FDA's technical staff interfacing with drug development sponsors, we begin to develop an annual list of regulatory science research needs that are specific for antimicrobial products. We take all this input into consideration in helping the Office of Infectious Diseases develop the fiscal year regulatory science research priorities. And there are several mechanisms we use to collaborate with our external stakeholders. We share information through requests for proposals, through FDA's broad agency announcements, which I will talk about more in detail later, and through the ORISE fellowship opportunities that Michelle just spoke about. The request for proposals identifies the Office of Infectious Diseases fiscal year priorities and solicits research proposals that we will fund that year. We post this information on our research webpage as well as on the contracts websites and grant websites. The FDA's broad agency announcement is an open solicitation that lists our priority topic areas for that year, and our section is listed under topic area 2.4. And these include to evaluate potential innovations in clinical trial design for new drugs, to advance the science of in vitro animal model pharmacokinetic studies and/or real-world evidence studies, to evaluate strategies to enrich enrollment in clinical trials, and to advance the science of antibacterial drug susceptibility testing for antibacterial drugs that are a high public health priority. And I've included the current link for the BAA solicitation. We also recruit ORISE fellows through an open competitive announcement. And this year we plan to recruit 2 antibacterial drug resistance fellows with experience in epidemiology, data analysis, and microbiology. And I've provided the links for these 2 announcements. So in summary, if you're interested in applying for research funding that aligns with the Office of Infectious Diseases priorities, please submit your application through FDA's BAA. If you're interested in being a fellow and interested in infectious disease research, please check out our ORISE Fellowship link. And both of these sites are hyperlinked in the slide. All of the information that I've presented today and background on the CARB research program can be found on the Infectious Disease Research Activities webpage. In addition, I can also be reached if you have any questions regarding the CARB research program. Now I will discuss FDA's broad agency announcement on behalf of the Office of Chief Scientist. FDA solicits research and development proposals to support regulatory science and innovation, and encourages science and technology-based firms as well as educational institutions to participate in meeting FDA's goals for basic and applied research in strategic scientific priority to promote regulatory science. Through the BAA, FDA seeks to support advanced research and development strategies and therefore priorities that are aligned with the strategic science plan are updated annually. Contract awards are made to advance scientific knowledge to accomplish FDA's mission to protect and promote the health of our nation. The BAA is an open and competitive announcement, and submissions are accepted on a rolling basis throughout the year. The BAA is valid throughout the period from the date of issuance through the closing date. Amendments to the BAA will be posted when they occur. The evaluation is a 2-tiered process. Stage 1 is the review of the white paper and 1-page quad chart. The white paper should provide sufficient detail to allow evaluation of the concept's technical merit and its potential contribution to the FDA mission. A technical panel will evaluate both the white paper and quad chart, and if favorable, the offeror will be invited to submit a full proposal. Offerors whose quad chart and white paper did not receive a favorable evaluation will be notified by email regarding technical deficiencies and/or a lack of programmatic alignment. The second stage is the invitation to the offeror to submit a full proposal along with the cost proposal. The quad chart, white paper, and full proposal will be evaluated by a scientific review process based on the following criteria: scientific and technical merit, program relevance, and capabilities and experience. The final evaluation by the technical panel will be based on an assessment of the overall best value to the government. Awards will be made based on the proposed evaluation, funds available, and other programmatic considerations. And these research awards are made through a contract mechanism. If you have any questions, the BAA contracting officer and the program manager will be available to answer them at the end of the session. In addition, I've included the BAA email address in case you have any follow-up questions. Thank you so much.
Hello, and thank you for joining us today for the Small Business Association webinar focusing on FDA's research program within the Office of New Drugs. As Dr. Laura Jaeger has laid out in her presentation, our office, the Office of New Drugs, does an extensive amount of research as part of our regulatory review activities. Some of that research is conducted on data that we generate in-house, or it also involves research in the sense that we review data submitted from the outside and analyze it and draw conclusions from it to support our regulatory activities. My name is Chris Leptak, and I'm the director of CDER's Biomarker Qualification program. I'm going to spend the next few minutes highlighting how we use data to generate information to support a biomarker's use in clinical drug development. My disclaimers, as with many folks at the FDA, I cannot have investments in the products that we regulate, so I do not have any disclaimers to share. So let's start with some fundamentals, since many of you may not be familiar with biomarkers, or you may be, but not necessarily in the sense that we think about them as a regulator. So let's start with the definition. A biomarker is a defined characteristic that is measured either as an indicator of a normal or pathogenic process, or as a response to an intervention. We think of biomarkers very broadly at the FDA, although the lay community thinks of them maybe as something that's measured in a body fluid, which is in fact a type of biomarker. We also can look at biomarkers from radiographic images or physiologic studies. So there's many different types of biomarkers that we can use as part of aiding drug development. It's important to emphasize that what a biomarker is not is how a clinical assessment of how a patient feels, functions, or survives. That contrasts it from clinical outcome assessments. This is important because when we look at a drug that potentially is going to be approved for marketing in the United States, that approval is based on clinical benefit, and clinical benefit is related to how a patient feels, functions, and survives. And although a small subclass of biomarkers, namely surrogate endpoints, can predict how a patient feels, functions, and survives, the majority of our biomarkers are not used in that way. That does not mean that they do not have value. In fact, they do, and I'll be highlighting some of those uses in subsequent slides. When we think of biomarkers, there's many different communities that do use biomarkers beyond us as regulators. For instance, there's the clinical community who may be using biomarkers to assess individual patients and trying to make good healthcare choices for them. Likewise, the academic or scientific communities are studying them in a more laboratory environment, trying to figure out the underpinnings of disease and what causes disease or how we might target it or treat it. Although there's many different communities that do use biomarkers, there's some considerations that we as regulators need to look at to figure out if a biomarker has a particular utility. One of those is reproducibility of the data. Although there's a lot of publications around biomarkers and their potential uses, it's a challenge in that many times the data from one lab to another is either studied in different ways or it's not consistent in its overall conclusions. And that reduces the generalizability of that data for us as regulators. Likewise, a biomarker needs to be adequately measured by some technology or platform that has been studied so that we can reliably use that information and interpret it so that the differences that we see are not related to its measurement platform, but rather than the biology behind the differences that we're witnessing. And then finally, we want to know that the biomarker is feasible in both a clinical trial setting and also if the drug should be approved, that the clinical community can use that biomarker and understand its use. I also want to draw your attention to a resource that's available. It's called the BEST Glossary. It's on the National Library of Medicine website, and the link is included here on the slide. This was a resource that was generated between FDA and NIH to establish a taxonomy of terms and consensus definitions so that when the different stakeholder communities are speaking of biomarkers, we're doing so with the same language and intent. So I highly encourage you to look at this resource if in fact biomarkers are part of your area of interest. In the glossary, biomarkers are broken down into multiple different classes based on how they're used. These classes are listed out here. I include them, but I think especially for those of you who may not be biomarker researchers or are knowledgeable about this field, I think the easiest way to understand these different classes is through a diagram on the next slide. So ultimately, if you think of a person, there's got to be a normal state, and that's what is depicted here in the diagram, the normal physiology. So as organic beings, we certainly are going to undergo change as part of our routine, both on a day-to-day, week-to-week, month, and then throughout our lifetimes. So one of the things that you need to establish when you're trying to figure out if a biomarker has a particular use is understanding what is that normal range or variability in a quote-unquote normal state, and also figuring out what is the variability within a person over time, between people of different ages, of different sexes, of different ethnicities. So there's a large part of study that's necessary to kind of figure out whether or not your biomarker has use in an abnormal state or condition, or in our case, a disease. Over time, our normal physiology changes. Some of those are detrimental and ultimately can result in altered physiology. Ultimately, then, if you get to a clinical disease, which is a constellation of signs and symptoms that's recognized by the clinical community, then you get into a new set of classes of biomarkers, namely diagnostic, monitoring, and prognostic. Diagnostic here kind of falls into two camps. One, the biomarker in and of itself is necessary and sufficient for the diagnosis of the disease, and that you don't need other information. More likely than not, we also think of diagnostic as a lowercase d, if you will, in that the biomarker information is diagnostic when it's used in combination with other signs, symptoms, or even other biomarkers. So it's one component of the overall diagnosis. Once a person has disease, especially if it's a chronic condition that waxes and wanes over time, you may want to monitor the disease to determine the severity at a given point in time, whether or not that condition has changed in some ways, that you may want to go to a different class of agents. But overall, you want to kind of see what the status of the disease is in the person that's being studied. And then finally, there's another class of biomarkers known as prognostic, where for a given disease condition, especially if that condition lasts over time, sometimes during exacerbations or as the disease severity worsens, there's an event in the future that we want to try to prognosticate because it's of importance. So an example here would be if you have an asthmatic patient, some biomarkers are indicative of the likelihood of a patient having to go into the hospital and to get more extensive care. So that would certainly be something of interest. Once you have a therapy in play where you're trying to treat or mitigate the disease, you get into the remaining classes of biomarkers. Here, where we're trying to change the physiology of the condition, we may be getting into the pharmacodynamic response class, predictive or safety. Pharmacodynamic is saying that we're targeting a component of the biologic pathway, hoping to either prevent its progression or perhaps even improve its overall trajectory. Sometimes predictive biomarkers are used when a given patient population is heterogeneous and we're trying to identify subpopulations that are more similar either in their biology or in their presenting phenotype, which can be beneficial for subsetting patients for trials. And then finally, safety biomarkers, in that therapeutics many times have off-target effects that can impact negatively different organ systems. And so we want to study those safety profiles. If in fact the therapy either prevents or improves the overall condition, we can then get more into the response class of biomarkers. And then finally, a very small subset of those response biomarkers, if in fact they predict clinical benefit, then we're talking about surrogates. And again, that's a sort of small subgroup of the overall biomarker classes. From a regulatory point of view, when we think of biomarkers in a drug program, namely in a clinical trial, we try to define what the biomarker's use is so that we clearly understand what data would be needed to support that use. To define the use, we have what we call a context of use, or a COU, which includes the best biomarker class that we just went through, and it also then describes how the biomarker impacts the clinical trial design or the overall drug development program. Listed here are multiple uses. They can be brought to play as part of inclusion-exclusion criteria for clinical trial enrollment. They can be used to monitor patients for safety concerns, and then they could also be used as an endpoint to determine if that therapeutic compound was effective. So these are many uses, not extensive, but some of them that are listed here. And at the bottom includes what a context of use description might look like for total kidney volume. When you're developing a biomarker, we've found over time that there are components of an overall program that are important to consider as independent elements as part of your overall project plan. So for us, we always want to start with the need statement. What is the need in drug development? Is it to try to reduce patient heterogeneity? Is it to try to have better markers of safety or toxicity? So it's nice to have what your goal is up front, and we call that the need statement. From there, we get into the context of use that we just described, that based on your need, we think that this biomarker can be used in this way. So it's the class of the biomarker and what the biomarker is addressing in the clinical trial context. Then, depending on how the biomarker is used within the clinical setting and the patient population, you can then talk about the benefits and risk of the use of the biomarker. And then that gets into the type of evidence that we want to see. Ultimately, in a biomarker development program for regulatory use, some of the components of the evidence include the analytics, looking at the performance of the measurement technology or platform, our statistical analysis plan, so that we can clearly look at potential areas of bias and know that when we make an interpretation that we can have great confidence in it. I mentioned that when you're looking at a biomarker, the measurement method or platform is very important. That can be broken down into two broad types of information. One is analytical validation, the other is clinical validation. Analytical validation just says that you can reliably and reproducibly measure whatever you're interested in our case, the biomarker. The clinical validity is a bit different. It is, not only does it say that you can accurately and reliably measure it, but that measurement and change that you're assessing has biologic or clinical meaning. So both of those, the analytic and the clinical validation pieces, are important. The program by which we qualify biomarkers at the FDA was laid out in the 21st Century Cures legislation that passed back in the end of 2016. In that legislation, it made FDA, established for FDA a very formal process for biomarker development, and it put us more collaboratively and proactively involved in the process, which we find after several years at this is a very good thing. As laid out in the statute, qualification involves the submission of 3 different submission types: the letter of intent, the qualification plan, and the full qualification package. In addition, the statutes put forward the transparency provisions, whereby we make public information about the submissions that we receive. Likewise, we make public our recommendations towards that project. Because this information is out in the public sphere on our website, it really has, a result has led to a shared learning culture where people can look at the types of projects that we receive. If it's in an area that's similar to something an outside party is interested in, they can learn not only from what other groups are doing, but also the advice that we give, and then incorporate that into their own projects. And what we found that even in a short period of time, the quality of our projects are steadily improving because people are learning from each other as we're going through this process. If you're interested in looking at what biomarkers we've qualified or what biomarkers are under development, the links for those are provided here. For each of the submission types, there's a distinct type of content that we look for. In the letter of intent, the submission is relatively small, 10, 15 pages, not long. It's not meant to be an NIH grant proposal. And the main components that we're looking at in the letter of intent is, what is the drug development need? Do we agree with that need, based on what we see on our side of the fence? A clear context of use to address that need, some information about the biomarker and that support its purported use, and then finally some summary level information about the measurement platform. The letter of intent is not a data review process, and that we just want summary level information at this point. If you get a LOI accepted, the next submission type is a qualification plan. And that plan is where you really put forward the detailed explanation of your overall development effort. It includes an assessment of the information that's known out in the public literature and in scientific journals. It identifies what your ultimate goal is, what the scientific knowledge gaps are to get to that goal, and what data you plan to provide or analyze to fill in those scientific gaps. It includes detailed information for analytic validation to the extent that it's known around the analytical performance characteristics. Likewise, we'd like to see a statistical analysis plan that we can then agree upon as part of the final data analysis. It's important that because this is a collaborative process, that in the qualification plan, you've thoughtfully addressed the comments and recommendations that we've previously provided and have incorporated those as part of that submission. The plan is basically a soup to nuts that once we can reach an alignment around what that plan is and have an agreement that it's a good one, then ultimately you, the outside party, can then conduct the plan, gather the information, do the analysis, do the interpretation, and then submit that to us in the final submission, which is the final qualification package, where it includes all the data, etc. One thing to point out, since this is a research seminar, that if you have a letter of intent that's been accepted into our qualification program, and this is true for ours and biomarkers as well as the other ones at FDA, that then makes you eligible for potential grant funding, and that the CURES legislation has given FDA monies each year that we are using towards supporting a new DDT grants program. We understand the value and importance of biomarker development, and not all parties have access to unlimited resources. And so we find that this is a way to help outside groups do additional biomarker development and then submit that data to us. And that's been proven to be pretty effective. We plan to continue that DDT grants program as long as the monies are available to us. Internally, we do a 3-tiered review. Once we receive a submission, the biomarker team will look at it, make sure it's administratively complete, that you've addressed any previous recommendations that we may have given, and that the overall descriptions of the different biomarker development components are well conveyed and understood. After that, we bring in our subject matter experts that include the clinical expertise, the statisticians, analytic experts, our clinical pharmacologists, and anyone else that has an expertise that's relevant towards the overall biomarker development effort. That group, along with the members of the qualification program team, put forward an analysis and recommendations that we then take to the deciding body, which is our CDER DDT committee. That committee is made up of senior level staff from throughout the center or their designees, and they're the ones that actually discuss and either approve or do not approve the submissions that we receive. Either way, whether it's accepted or not, the outside party receives recommendations of how they could either improve the development effort or to make certain amendments so that we can reevaluate the package as part of a submission in the future. Finally, I'd like to end my presentation by 2 slides on surrogate endpoints. Certainly there's a lot of discussion of surrogates because of their potential value in drug development. At the FDA, we have 2 important resources that I'd like to draw your attention to. The first is a table of surrogate endpoints that we make publicly available and update every 6 months. This was one of the deliverables that was part of the CARES legislation, and we've worked extensively with our clinical divisions to compile a list of surrogate endpoints that are acceptable to either accelerated or traditional approval. They can be found at the website listed here. What you'll find when you look at the resource is that there's hundreds of surrogate endpoints that can be used for various disease and patient population combinations. And so I highly encourage you to look at that. I think it's a good resource, and it helps for external folks to understand how open we are to the use of surrogates when it's appropriate. So please take a look at that resource if it's of interest. Likewise, if you're a drug developer, we do have a new Type C meeting process for novel surrogate endpoints that can be requested. We're here if you have preliminary clinical information for a novel surrogate, one that has not been used previously for a drug approval of any type. Then you, as part of your meeting package, you provide detailed information about that biologic surrogate to us. We have an internal discussion. We take it to the DDT committee that I mentioned previously, so we get senior level input, and then we can provide you feedback around your overall proposal. Included as part of the meeting package or as a content outline with the elements outlined here to help you tell us why you think that the surrogate is appropriate and what you know about it to date, so that we can give you good recommendations moving forward. So I hope this has given you a bit of a sense of how we can use research data. In our case, most of it is generated externally, and then analyze it internally to help us make good regulatory decisions. In our case, develop biomarker drug development tools to support the overall drug development platforms. Thank you for your time.
Thank you, Thushi and Christopher, for those great presentations. If you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. Our first question will go to Thushi. Where can we learn more about the ongoing CARB study?
So all of our CARB research studies that we award, we list them on the Office of Infectious Diseases research webpage. And we update this list every year, and we're in the process of updating it. So if you just go to our webpage, you should be able to find everything we've awarded so far.
Thank you for responding to that question. Our next question will go to Christopher. How often are projects terminated early? If this does occur, does FDA find another PI to complete the project? Is it reopened or simply sunset?
That's a very good question. To be clear, that the projects that we receive are initiated and owned by the outside parties that are conducting the research. That said, biomarker development can be a process that occurs over several years. And as such, different groups either lose funding or they change the members of their consortium. And so it's not unusual for us to see a project that transitions from one group to another. That said, that transition is initiated and takes place by the outside party, not by us. And in terms of the question of how many of them sunset early, actually a relatively small percentage. Development does occur over a period of time, but it's usually fairly rare that we get someone who will start the process and then withdraw it. And even in those cases, more often than not, it's because of a lack of funding and resources rather than that the science isn't cooperating.
Thank you for responding to that question. We have another question for Chris. How can patient-reported outcomes data be used to select or qualify a biomarker?
So one thing to keep in mind is that when it comes to drug development tools, or DDTs as we refer to them, they come in different varieties. My presentation focused on biomarkers, which is one type of drug development tool. The clinical outcome assessments are a separate class or type of tool, and they have their own qualification program. It is possible that depending on a person's proposal, and if it's a composite tool that includes both biomarker and COA information, that both of our programs will work together on our side of the fence. That said, the two programs are in fact distinct and separate. In terms of the DDT grants program, the funding is not just to support biomarker development, but it's drug development tools more generally. And so it is possible that a COA program that also has an accepted LOI is also eligible for funding. And I would just encourage you to just Google DDT FDA, and you'll certainly will get hits that will bring you to web pages both for my program and for the COA program.
Thank you for responding to that question. The next question is addressed to Thushi. How do I start a Stage 1 application?
So the first part of applying for funding under CARB is to go to our priorities list, which is listed in section 2.4 of the broad agency announcement. And I think at the end of the broad agency announcement is a section on how to apply for funding, and it will have both the Stage 1 and the Stage 2 application process.
Thank you for responding to that question. We have another question for Thushi. When will the Office of Infectious Diseases post information on funding opportunities?
So the link for our funding opportunities will also be listed in section 2.4 in the broad agency announcement. And funding opportunities can also be found on our Office of Infectious Diseases webpage. And this should be posted sometime this month.
Thank you for responding to that question. Our next question goes to Chris. For the DDT grants program mentioned in your presentation, can you provide any additional information?
So if you go onto our program's website, there's information that will be posted about the grants program. And then that's true in general. Pretty much anything that you need to know about how to qualify a drug development tool, you'll find an extensive set of resources there. So I highly encourage you to look if that's of interest. The announcements for the DDT grants program is annually. Usually the announcement will go out sometime in the December-January timeframe. We'll certainly put out information on our website when that occurs. Do keep in mind, though, that you have to have an accepted LOI in one of the qualification programs in order to be eligible to apply for that funding. We do not fund just research in general like the NIH might. Because you have to have an accepted LOI, we reach out to the points of contact for the projects that are in our respective programs, and then work with those folks directly to alert them that, in fact, the funding is available. I understand there was also another question about the amount of the funding. In general, depending on how much budget we have each year, and again, we'll continue to offer the program as long as the money is there. In years past, it's been up to $250,000. And that money is then used to help further develop the tool and to get the outside party to the next submissions as part of the three-step process.
Thank you for responding to that question. We've got another question for Chris. Can we submit an LOI for cancer biomarkers and drug development?
The simple answer is yes. As you'll see if you look at the ongoing projects that are on our program on our website, the projects span pretty much most of our clinical divisions, and certainly cancer is one of them. So yes, we would accept an LOI in a cancer space, absolutely.
Thank you for responding to that question. We got another question in for Chris. Could you describe the steps needed to get an approved LOI that allows for grant proposal submission for biomarker development?
That's probably more than I can cover in a minute or two here on the website. Again, I highly encourage you to look at the website that's listed in my slide presentation. And again, you can just Google FDA Biomarker Qualification. In addition to the resources, it'll also provide information of how you can reach out and contact us. So I would suggest that you do that first. And then if you still have additional questions, just please reach out.
Thank you for responding to that question. We have just a few moments left. Oh, well, we only have 30 seconds left. I don't think we have any more questions that we can do in the next 30 seconds. I would like to take this opportunity right now to pass over to Shayla to make some remarks.
Program is low. Hi, good afternoon. This is Shaila Shahid. I manage the broad agency announcement program across the agency, which is what Thushi had kindly presented on our behalf. FDA BAA inbox address was also added by Thushi on the presentation. Please feel free to ask questions, and we'll be happy to assist you as you kindly think about the announcement. Thanks.
Thank you to all of our presenters for your very informative and timely talk, and also for responding to the questions that came in.